higher biology. Doing replication, okay? So basically the idea is your two strands of DNA, which are, they form two new strands, okay? Okay? So each parent strand acts as a template for a new strand. Each parent strand each strand acts as a template. Okay? And it uses lots of nucleotides. Okay? Plus ATP, plus enzymes. Okay? So yeah, nucleotides, ATP and enzymes. So you have, remember it's A, T, sort of G along here, okay? And then you get fork forming, okay? The DNA is unwounded, okay? So this is our the DNA is unwound, okay? You get primer, 3' end, okay? 3' end of DNA, okay? That's the 3' end, that's the 3' end, okay? Just remember this, the base is A, T, C, G, okay? You get nucleotides coming in, okay? Which is a phosphate attached to a pentose base, okay? A pentose sugar and a base, okay? That's a nucleotide, okay? So nucleotides coming in, individual ones, which is base, sugar and phosphate, okay? And they basically, so you get you get individual nucleotides coming in. So the DNA, that's polymer, or the enzyme called the polymerase, DNA polymerase. I said enzymes have optimal temperatures and pHs, and they take a product, they take a substrate and turn it into a product. In this case, the substrate is the nucleotides with this enzyme. So the nucleotides are an enzyme. Sorry, the nucleotides are the substrate. The enzyme acts on them, and joins them together, okay, to make the new DNA strand, okay. So yeah, I'll repeat that. The nucleotides are the substrate, the enzyme takes the nucleotides and joins them together and makes a new DNA strand, okay. So this is the complementary strand of replicated DNA. This is template. And this is the complementary strand. Of the DNA, okay? So these get joined together. This is called, this strand here is called leading strand, okay? Okay? And the other strand, what happens to the other strands is a little more, slightly more complicated. You get, so if I can, so what happens in this strand is that you get, so you got, so this strand goes on, it's the leading strand. The other strand is made, it's done a bit differently. You get, okay, so you get, this is called the lagging strand. Okay. The direction of replication is actually that way, okay? That's the direction of it, it's going that way along. Okay, the DNA polymerase is here. That's the DNA polymerase, okay? The lagging strand is made of primers again, which, so you got 3' end, okay? And then each one has lots of nucleotides joined to it, okay? They don't actually form the shape, I'm just showing you the nucleotides get attached, okay? So replication fork DNA polymerase primers. It's called lagging because it's sort of lagging behind. It's made in bits rather than one continuous length. So you get primers coming along being joined, and then you get another enzyme coming in called a ligase, which joins the nucleotides, okay? So it joins the fragments. Okay, so ligase enzyme joins the fragments forming. So this is, I said that was the, sorry, that's the 3' end, and that's the 5' end, okay? So that's the 3' end there, okay? So this is called lagging because it's made of bits, and all these primer and then nucleotides get attached, and then the nucleotides, so actually, although drawn like this, you'd have, okay, and then you have, sorry. Okay, imagine these are all separate nucleotides and not actually, they're all joined together. I'm trying to put tiny gaps in so you can see they're separate, okay? And then they get joined together, okay? Ligase joins them. Okay, so that's why it's called lagging. So that's what's going on. They aren't actually necessarily coming off, it's just trying to at least show you how it works, okay? So yeah, that's the basics of DNA replication. So it kind of uses ATP, nucleotides, ligase, DNA polymerase, plus DNA with a help. Nucleotide ligase DNA, okay? So that's the basic, and you get many replication forms in DNA, in a DNA so in the long molecule of DNA, you get you'll get this occurring, you'll get sort of so these are replication bubbles, okay? These are replication forks. Okay? And this is a replication bubble. Okay, so that's kind of how it works. There are also other enzymes as well, but you aren't meant to know that at this point. So yeah, the lagging strand, this is called, when it's the leading strand, it's continuous replication. This is called, so that's continuous on the leading strand, but if the lagging strand is discontinuous, okay? The replication of DNA, the lagging strand, is discontinuous. The leading strand is continuous, this strand is discontinuous, but it's made of lots of sections, okay, that join together. So that's how DNA replication works. Good afternoon.